Today, let's explore how geneticists understand where enhancers are located in DNA sequences. And to start, let's consider a strand of DNA, which is during interphase sort of unwound inside the nucleus. And I also want to remind you that DNA is packaged into proteins, histones. And the DNA can either be tightly packed, but in other regions it can be loosely packed. And this distinguishes two types of chromatin structure: heterochromatin, which is the densely packed type, and euchromatin, which is where the DNA is more loosely associated with those histone proteins. And also remember that heterochromatin is repressive for transcription, which means that when DNA is very tightly bound to these histone proteins, it's hard for other proteins like transcription factors to access genes and to start transcribing them. So it tends to be the case that transcription start sites and then their promoters, and also enhancers, which is where transcription factor proteins bind, tend to be near, in three-dimensional space inside the nucleus, the transcription start sites of genes that they regulate, because these are physical interactions between transcription factors and things like, say, RNA polymerase. So red are transcription factors. Those transcription factors bound to their enhancers near the transcriptional start site in three-dimensional space are things that, say, stabilize RNA polymerase at the transcription start site so that transcription can begin. And transcription can be inhibited when those coding regions, which we can schematize in yellow, so let's say this is the coding region of that gene, those also need to be in euchromatin for all of these proteins like RNA polymerase to pass along the DNA strand. So it tends to be the case, to reiterate, that both enhancers, promoters, and coding regions of genes are in euchromatin when those genes are being actively transcribed. We should also note that if we blow up one of those histones, each nucleosome, which is one histone with usually a couple of turns of DNA wrapped around it, is made up of 8 different proteins. So in this ball, we can only see the 4 that are facing us. And those 4 proteins are histone H2A, H2B, H3, and H4. So the other 4 are the exact same 4 proteins on the back side of this globular set of 8 proteins. So this is an octamer. And each of those histone proteins has, at its amino terminus of the amino acid sequence of those proteins, some lysine residues. And in this case, I'll say that the 9th amino acid in the histone is often a lysine. So that's K9, a lysine at position 9 of the amino acid. And those lysines can be chemically modified, and this is called epigenetics. And when histone lysine 9 has an acetyl group added to it, that is a mark of euchromatin. Again, that's the chromatin structure where the DNA is loosely associated with histones. And so it's often the case that when histones are acetylated at lysine 9, that's a mark or an indicator of euchromatin. So for example, in this case, we would expect these histones in the euchromatin to have that K9 acetyl mark, where other histones that are in heterochromatin do not. So to summarize again, there are a few different characteristics of euchromatin. This open chromatin structure tends to contain enhancers, promoters, active genes at particular points during the life of an organism and during the cell cycle, of course. And the histones that associate with these regions of open chromatin tend to have histone lysine 9 acetylated. And last, the enhancers, promoters, and coding regions tend to be physically near each other in three-dimensional space inside the nucleus. And another interesting fact about the nucleus and its organization is that chromosomes during interphase, so now I'm going to switch color coding. I'm just going to use black to depict the nucleus. I'll use blue to draw a single chromosome. And I will use red to denote the position of a transcription start site and an enhancer, which again is a DNA sequence that a transcription factor will bind to regulate expression of that gene. So that blue chromosome isn't just randomly organized or arranged inside the nucleus. In fact, some parts may be disorganized, but other parts will always take on the same shape. In this case, that enhancer sequence is always near the transcription start site in three-dimensional space inside the nucleus. So there's a common feature here. If we look at multiple nuclei, we'll always see that the enhancer is close to the transcription start site when that gene is being transcribed. But if we looked at other parts, so let's draw that interaction as yellow. So those two parts of the chromosome are always near each other. But if we look at other parts of the chromosome, we would see that, for example, here, in the middle of the chromosome, there's a region that's in one nucleus. Those two regions are close to each other. But in the other nuclei, those regions are far away from each other. So this is one way, if we had a way to figure out the three-dimensional conformation of chromosomes inside the nucleus, we could look for associations where more frequently than by chance, so these indicate sort of random interactions, that sometimes they're far apart, sometimes they're nearby. That's sort of what we would expect by chance if the chromosome was randomly folded inside the nucleus. But if we looked at different parts of the chromosome, we would see that there's an increase in the probability that we'd find those two regions of the chromosome near each other. And that's another way to identify potential enhancers of genes, is we look for regions that are statistically associated with each other more frequently than we would expect by chance. And if one of those regions is a gene, then it's likely that the other region is an enhancer that regulates it. That brings us to three different methods that can be used to identify potential enhancers. The first one we're going to look at is the oldest, called ChIP-seq, for chromatin immunoprecipitation sequencing. And the way this works is we look again for places where the DNA is either tightly associated with histones, heterochromatin, or loosely associated. with, again, the idea that loose association will either indicate the position of a transcription start site or an enhancer. And remember that each of these histones in the euchromatin has a particular lysine 9 acetylation on its N-terminal tail. So what happens in ChIP-seq chromatin immunoprecipitation is that what we're looking for is we want to get the DNA sequence of the genome of the chromosome that's associated with acetylated histones, because those acetylated histones are in euchromatin. So this is a way to figure out what is the DNA sequence that's associated with euchromatin. So that's the goal here. What is the euchromatin DNA sequence? So this is not a targeted approach in the sense that we're not asking a specific question like, is a particular part of the genome in euchromatin? This is a discovery-based approach where we get the sequence of all of the DNA that's present in euchromatin at one specific point in time in a tissue in a cell. So what happens in ChIP-seq is the DNA and proteins are chemically cross-linked to each other, so the DNA can't be separated from the proteins at present, with usually something like formaldehyde. And at that point, an antibody that's specific to acetylated lysine 9 on histone 3, so anti-H3 lysine 9 acetyl, is added. So that antibody physically binds to the histone protein, which is chemically bound to the DNA that it's presently bound to. Then the DNA is fragmented, so the DNA is broken into bits randomly, essentially. And then those antibodies are precipitated. And I'm going to diagram this with a magnet, although that's not actually, well, it could be one way that the antibodies are removed. But that basically attracts then all of those antibodies, purifies them from the rest of the DNA, proteins, and so forth. So what we wind up getting is DNA sequence, little fragments of it, that were associated with histones that had lysine 9 acetylation on them. And those short DNA fragments are then deep sequenced, and that then gives us the sequence that was associated with euchromatin inside this cell at whatever point in time during the cell cycle and during development this cell was analyzed. Another method of doing essentially the same thing, figuring out what are the DNA sequences or what are the parts of the genome that are associated with euchromatin, is a technique called ATAC-seq for an assay for transposase-accessible chromatin, followed by sequencing. And this is sort of similar. If we have the position of a transcription start site and its enhancer, I think I might be switching up the color coding here. The enhancers will tend to be more in euchromatin than in heterochromatin, and the same for the coding region of a gene. And other regions will be more in euchromatin, where those histone proteins are more tightly bound to the DNA. The idea here is that there's a short piece of DNA, TN5, which is a transposase, a type of jumping gene. And in this case, so that green is DNA. It's been chemically modified to include particular DNA sequences in yellow that are going to be primer sites for polymerase chain reaction amplification. And what happens during ATAC-seq is, as usual, cells are collected at a particular point in development, a particular point in time when the scientist is curious about where the enhancers and genes are. And that transposon is then activated. And what the transposon does is it, again, is a jumping gene. It actively inserts itself into DNA. And the concept here is that it can't insert itself into heterochromatin because the DNA is too compacted and tightly associated with proteins, the histones, but it can insert itself into euchromatin. So what happens then is after transposase treatment, that transposase has integrated itself in the genome wherever there's euchromatin, and it goes along with those little primer sites. And those primer sites then allow PCR to be conducted. So let me diagram that. What happens is PCR primers are used to target, for example, a forward primer in that transposon, and a primer target in that transposon. So essentially, PCR is conducted that amplifies lots of copies of the DNA that's in between the transposons. And so that makes lots of copies of those regions of the genome between the primers that were inserted into the euchromatin because they are primer sites that are in this engineered transposon. So then, those PCR products are sequenced, and that gives us the sequence of the DNA in between the transposon insertion sites that are part of euchromatin in this chromosome. The last technique to talk about right now is a technique called 4C-seq, and that is for chromosome conformation capture circular, which is why it's called 4C-seq. So what we're doing in this type of experiment is, again, looking at that three-dimensional structure and trying to figure out what parts of the genome are close together in physical space. Because, again, those could involve a transcription start site of a gene and also the sequence of its enhancer, a spot where a transcription factor will bind and regulate or interact with the transcription start site and the proteins that are bound there in the promoter. So once again, the question is, how do we figure out for a particular nucleus and for many nuclei where the regions of the chromosomes are that are near each other in three-dimensional space? So this is similar to a technique that we've looked at before, ChIP-seq, where at a particular point in time, the DNA and the histones are cross-linked with something like formaldehyde. And then what happens is the chromosomes are broken randomly, and those pieces of DNA then are ligated together. So because in three-dimensional space, these regions of the chromosome are sort of locked in place with all of this cross-linking, when the chromosome is then broken, there are free 5' and 3' ends of these parts of the chromosome that are free to float around in space. So what happens is those free ends will eventually base pair with each other. Well, the 5' and 3' ends, they don't base pair together, but the 5' and 3' hydroxyl and phosphates come close together, and they're ligated. So basically what this winds up making then is a circular molecule where the enhancer is over here and the transcription start site is over there. And if we showed where those chromosomes broke, it would be, for example, there, those two ends ligated together, and the same for the other two breakpoints. So in other words, that breakpoint is here, stuck together with the end of that breakpoint. And the same up here, that end where the chromosome was randomly broken during this process, there, ligates to that end there. So at this point, we have a couple of circular molecules of DNA, and they both represent DNA that was in physical proximity to other parts of the chromosome, because that's how this cross-linking works. It only cross-links or covalently connects parts of the chromosome that are physically touching each other, essentially, inside the nucleus. So what happens then is these circular molecules of DNA are sequenced. So that gives us the DNA sequence of the region of the chromosome that was physically near another region of the chromosome, or a different chromosome even, at this point in development of this organism. So finally, let's take a look at how these data are analyzed and how we interpret the data that come out of all of these different sequencing experiments, like chromatin IP sequencing, ChIP-seq, ATAC sequencing, and 4C sequencing. So in this case, I've drawn 3 representative nuclei, again with a chromosome. In this case, I'm indicating it goes from 1 to 100 base pairs long, and the position of one gene. So the chromosome may be arranged randomly, or it might not be arranged randomly. And again, that's what we're trying to find out. Is there a region of this chromosome, for example, that seems to be potentially an enhancer, a regulatory element that's found far from the promoter or the transcription start site? So we can see here that I've drawn it, as we've seen in the past in this presentation, that there's one region of this chromosome near the start of the chromosome that's frequently found in close proximity to the transcription start site. And so with any of these sequencing-based techniques, what we're going to find is we're going to get a lot of DNA sequence that contains the enhancer at the start of the chromosome and also the transcription start site, because both of those are found in euchromatin. So from ATAC-seq, we would find lots of sequences where that transposon inserted in those regions of euchromatin. From ChIP-seq, we would find lots of sequence from these regions, because those euchromatic regions are associated with acetylated lysine 9 on histone 3, so we'll get lots of sequence from those, but not sequence from any of the other regions of the chromosome. And from 4C-seq, because these regions are close together in 3-dimensional space in the nucleus, we'll see lots of sequences from them. So the way that these sequence data are plotted is the same for all of these techniques, so we only have to talk about it once. The x-axis on this graph is always going to be chromosome position in base pairs. So in this case, it's chromosome from nucleotide 1 to nucleotide 100. And the y-axis is something called read depth, and it can be measured numerically in various ways. But the idea is that the more sequences we get of each nucleotide on the chromosome, that tells us those were regions of euchromatin, or regions that were in contact with each other in 3-dimensional space. So in this case, for example, in any of the 3 techniques we've discussed, will we see any sequence of chromosome or nucleotide 100? Well, it's not in euchromatin because it's not a gene and it's not a promoter or an enhancer. So it will be in heterochromatin most likely, so we shouldn't see much sequence from it. It's also not in contact with other regions of the chromosome, so we wouldn't see sequence from this in 4C-seq. And if it's in heterochromatin, no transposons would have inserted there, so we won't get any DNA sequence associated with that nucleotide. So in this case, if we plot in blue, the read depth, the number of DNA sequences we get out of any of these techniques, would be low. And where it would be high is in regions where, for example, in ChIP-seq, the enhancer is in euchromatin and so is the gene. So let's talk in blue, let's say that's ChIP-seq. We would see lots of sequences come from the regions near the start of the chromosome, That would be the enhancer. And we would also see, but then it would drop off, because as we move away down the chromosome, this is probably, in ChIP-seq, this is probably heterochromatic if it's not containing a gene or an enhancer. So we would see that for the next part of the chromosome, there's very little sequence that comes, maybe none, maybe very little. And then once we reach the transcription start site and the promoter, we'll see that spike. And maybe we'll see spikes for exons that are in euchromatin, and then it will drop off again. Now, in one of these three cells, there is a random association between two other parts of the chromosome, but it's only present in one of these three example nuclei. So when we get to that part of the chromosome, we might see a tiny peak, but not as tall as some of the other regions. And we'd see essentially the same thing looking at another technique. So in ATAC-seq, for example, we should see essentially the same plot, because again, ATAC-seq is based on identifying regions of euchromatin where transposons can insert, and where the transposons insert in the chromosome, we will see peaks. So in this example, in these three nuclei, in my mind, the only regions of euchromatin are an enhancer near the start of the chromosome and the transcript start site and the coding region itself. So in that case, we might expect to see lots of sequences come again in ATAC-seq near the enhancer where there's euchromatin, and then it will drop off. We'll get a sort of a baseline background amount of sequence from other regions of the genome where transposons might rarely insert even in heterochromatin. And then we'd see a spike in the promoter transcript, and then we would see it drop off again. And in this case, when we get to the rare cell that might have some other sort of three-dimensional structure, we wouldn't necessarily see that association because this could still all be heterochromatic. So in that case, actually, I should go back and correct myself. If it's heterochromatic, that means we wouldn't have seen a ChIP-seq peak there, because ChIP-seq also deals in heterochromatin state. So we would see something like that for ATAC-seq as well. Now it's the 3C-seq, or 4C-seq rather, that's looking for the chromatin conformation, the three-dimensional associations where we would see that peak in that one random nucleus where there would happen to be a rare three-dimensional structure of the chromosome that brought two regions of the chromosome together that weren't a gene and a regulatory region. So in this case, in 4C-seq, we'll see a peak associated with these regions of the genome that are close together, and that's the only regions that we're going to get sequence from. So we won't see as much sequence over here because, again, 4C-seq depends on chemical cross-linking and ligation and circularization of only parts of the DNA that are physically near each other in three-dimensional space. So we'll see that come there from that nucleus. From this nucleus, we would get some sequence there. And here is where that random association between far away, linearly far away parts of the chromosome will randomly close together. We'll see some sequence there in ChIP-seq. So in this case, we get that sequence near the start of the chromosome, then we get very little sequence. We'll see sequence in the promoter, the transcription area, the coding region. And then we will see that little tiny blip of peak there where there's a random association between two different parts of the same chromosome. So overall then, these three different methods are sort of frequency-based look at which regions of a chromosome are in euchromatin, that's ChIP-seq and ATAC-seq, or are physically close to each other in three-dimensional space, and that's 4C-seq. So there are different ways to answer the question of what regions of the chromosome are in euchromatin and physically close to each other, and those tell us most likely where enhancers are located. So in closing, we will expect that where there's a transcription start site and a complementary or coding DNA sequence, a CDS, the coding region, we will definitely expect to see euchromatin. So that will be peaks for ChIP-seq and ATAC-seq. And what we'll see is there will be regions of the genome that aren't known to have a gene. So if we see a peak in any three of these techniques, ATAC-seq, ChIP-seq, or 4C-seq, if there's no gene there, why would it be physically close to another region of the chromosome and euchromatic? Those pieces of information together suggest, they don't prove, but they suggest that that region could contain an enhancer.